Hi all, welcome back. There's going to be two videos this week. The first of which is this one you're watching right now on data analysis and regressions. And I'm posting this because it relates to both the Diffusion lab write-up that you're working on right now, and it's also going to relate to the Daphnia lab part 2 that you're going to start this week in lab. You are now watching part 1, which, as I just mentioned, is related to Diffusion part 2 and your write-up. I like to call it linear regression, or how I learned to stop worrying and love the graph. Our next video in part 2 is going to be on dose response, which is going to show you an authentic biological use of bioassays, and again, using regression to answer some questions. One question you might ask is, why do we use regression in the first place? And the answer is that regression analysis is a form of statistical analysis. We'll be getting into statistical analysis using R shortly after Thanksgiving, but it's nice to look back at the regressions and say, oh, we were doing something statistically even before we learned about that. And it's like asking a question, is the variance in a y-axis variable in an experiment that we might do on a graph explained in some way by what we did in our experiment by changing the values in the x-axis variable, right? We change x-axis, we're looking to see if there's any variance in the y-axis. In this week's lab, we asked a question. If we change the surface area to volume ratio of an agar block, does that really relate in any way to the change in the rate of diffusion that we might see? Does X influence Y? And it's really the same question that we will end up asking by using a set of t-tests later in the semester, where maybe we would like to ask the question, is there a significant difference between the rate of diffusion in a 1 inch by 1 inch by 1 inch cube versus 64 smaller quarter inch by quarter inch by quarter inch cubes? And the biggest benefit of a regression is that it can provide us with something that's descriptive, and it provides us with a model that has predictive power to describe some sort of relationship. I want us to look at a silly example because sometimes it becomes important to consider a question. What does the null hypothesis look like? And how do we compare our experimental data to this null hypothesis? Now, a null hypothesis, again, is an idea that comes from statistics. And it's really asking us the question, what would we expect to see if our experimental variable had no influence whatsoever on the data that we're trying to collect? In this example with Snickers bars, we're saying that the null hypothesis is that Snickers bars have no influence whatsoever on the number of jumping jacks that somebody might be able to complete. And if we look at this data right here, it's looking like that these numbers are pretty similar, even though we're asking people during individual trials to do their jumping jacks with no Snickers, after eating 2 Snickers bars, all the way up to 12 Snickers bars. And I suppose because we're getting kind of close to Halloween, maybe we'll think of these as a little fun size and not full-size Snickers bars. But our alternative hypothesis, if there is in fact a relationship between the two, would be that there is going to be a difference between Snickers bars and the number of jumping jacks that people would be able to do. Another way of saying this is that our null hypothesis would be that we would not see any change in our y-axis values, which are jumping jacks, as we change the x-axis values, which are the Snickers bars that somebody eats. And it would just represent a straight horizontal line. So if y doesn't change as x does, we might see something that looks a little bit like this. If we were to fit a line in between all of these data points, because they don't look like they're changing a whole lot, it would look like a straight line, meaning that every value, no matter how many Snickers bars we consume, seem to fit right around 225 jumping jacks. That means that there's no relationship between consumption and the jumping jacks, and that's exactly what our null hypothesis would describe. A regression line is a line that fits our data closest by finding the minimal distance from each point collectively. So on our graph on the left, as our x-axis variable increases, our y-axis data also seems to increase in a direct relationship. And the line that we have has minimized the distance from each point to our line in between. If we thought about this line as being, say, a yardstick, and this yardstick was being held up to the wall by a bunch of nails, that were each connected to rubber bands, this is going to be where it's going to settle, because all of these rubber bands are pulling as little as they possibly can to settle this yardstick at its particular angle. If we wanted to compare this relationship to the null hypothesis, then we would have to manipulate the yardstick and pull it into a straight horizontal line. And we can see that over here on the right-hand side and the left-hand side, the rubber bands end up pulling pretty tightly because our data points are further away from this line. So this would be the best fit for the null hypothesis, which looks like it's under a lot of tension. So our line is pretty far away from the null hypothesis that there is no relationship between x and y. And we could settle on a conclusion that this is a better fit. And now we have a slope of a line that describes a model of increasing y-axis variables that correspond to increasing x-axis variables. We can think about rubber bands when thinking about a best fit regression line and what makes one a good fit versus a poor fit. This model over on the right-hand side is a pretty poor fit for the data that we collected. So how do we know if it is a good fit? What do we look for to identify a good relationship, or a relationship between our experimental variable and our responding variable? We can get a bit of information by using the R² variable that comes out of Excel. So we've already learned how to produce a regression line by just right-clicking on a data point, adding a linear trend, and that provides us with the equation that includes the slope of our line that shows us our relationship, where presumably on this hypothetical data, we would say that for every Snickers bar produced, somebody is able to jump 11.7 more jumping jacks than having not had any Snickers. This R² value is an indicator of how much of the y-axis variable is related to what we changed in the x-axis variable. Numbers that are very, very close to 1 means that we have a perfect correlation. R, or the R² here, is a correlation coefficient. 1 means 100%. Another way of saying that is that R² represents the fraction of the variance in y that is explained by the variation in x. If there's no or little relationship at all, R² is going to be very small, or it's going to be very close to 0. If there's a strong relationship and the data has little bit variability, R² will tend to be very large, and it could be a negative number close to -1, or it could be a positive number close to +1. Sometimes we can have a strong relationship with a large amount of variability in samples, but here the R² is going to decrease because the variation decreases the certainty that x is directly influencing y. It could suggest that there are other variables that are currently acting on our experiment that are not the one that we are testing. We can see that graphically with two separate examples. In our top graph, we see that there's a relationship between x, Snickers bars, and y, jumping jacks completed. And at each one of our data points, our individual trials seem to be clustered pretty close together, meaning there's not a whole lot of variability here. Everybody is affected mostly the same way by Snickers bars. Our R² value in this particular graph is 0.93, which we can interpret as meaning that 93% of the change in people's ability to do jumping jacks is directly related to the number of Snickers bars they consumed. In the bottom graph, this one having been made in R, we see that there's a lot more separation in the data points. And because of that separation, we have a lot more variability. And even though we see the same relationship, our R² value is a little bit lower, which suggests that there is something other than Snickers influencing the number of jumping jacks that somebody can complete. So the R² is a great way to look at the correlation and the import of your x-axis variable that you are currently testing. This is important for your diffusion report, right? Because you're presenting your data in figures. None of us want to look at a big table of all of your data in a lab report. So please make sure that you're adhering to the requirements of a well-crafted figure from the Writing in Biology book and from last week's lecture videos. The formatting, placement, graph choice, your call-outs, the way that you create your figure captions and titles are all going to be graded and are going to make up a large portion of the grade for this report. Also remember, you're presenting two separate sets of results here because you looked at the rate of diffusion with relation to surface area to volume ratio. Those are the three experiments at the beginning of lab. You're also looking at how the rate of diffusion changes based on your tested variable. If you fail to adhere to the formatting guidelines, then you're likely to lose points in both sets of results, which would be unfortunate. So what does all this look like for the diffusion lab? So let's look at the very first experiment that we did, in which you dropped a 1 inch by 1 inch by 1 inch cube of sodium chloride containing agar into some water, and we watched as the conductivity rose in the bathing solution over time from 0 seconds all the way up to some maximum number. I think it was 600 seconds in your experiment. And then you were able to do a linear regression in Logger Pro, which spit out some numbers. Gave you a nice regression equation with a slope here that shows us that our average rate of diffusion was 1.2 microsiemens per centimeter per second of increased conductivity, which of course we can correlate to sodium chloride concentration. And what you might not have noticed when you did this in lab was that you have a correlation, in this case it's 0.96, that says that it's very likely that over time, through diffusion, we see an increase in conductivity and that we are able to calculate a rate from that. But that really doesn't answer any of the questions that we had, which is why we went and we did. Which is why we did this not once with just one cube, but a second time by breaking that cube into 8 equal-sized pieces, and then finally with 64 cubes, each time changing the surface area to volume ratio, but in each case asking how did the rate of conductivity or the rate of diffusion increase as we move through the experiments, and we got 3 separate rates. But that doesn't answer the question that we were really getting at, which was how does surface area to volume ratio, as your x-axis variable, influence the y-axis variable, which is the rate of diffusion. You already answered this in part 1 of the lab. Hooray, right? It was a 4-point question. Generate a graph to display the relationship between surface area to volume and the rate of diffusion of sodium chloride from agar cubes. And ideally, it would look something like this. So now you've made a graph that incorporated the data that you collected from the initial experiment that didn't answer your question directly and turning it into a graph that does answer your question directly. Because here, you're graphing the surface area to volume ratio that you calculated at the very beginning of the lab, and you're plotting that against the rate of diffusion, which represents the m value from those first regressions. Here's your 1 cube experiment. Here's the 8 cube experiment, and then finally that 64 cube experiment. And those two sets of graphs together, maybe you could combine the original traces from Logger Pro into a single graph, and then the second graph tell the story. How did I collect my data, and what does it mean? You'll also be doing something very similar for the new variable that you and your group tested that was different than the other groups. So that, I think you've got enough information now and enough understanding that you'll be able to answer and put together your lab report with a lot of care and telling a good story. So follow up with some announcements. Please do not forget Quiz 5, which opens up on Monday morning at 12 a.m., bright and early. And then 48 hours later, closes on Tuesday at 11:59 p.m. This quiz is going to be relevant to both the Daphnia lab as well as review. Bring a calculator, practice your dilutions and your regression equation calculations, right? Because you're going to be interpreting graphs and regressions. Your diffusion lab report part 2, the written report is due in lab this week by whatever means your ULI or faculty member requests. Not on Monday, but during the day of your lab. And then instructions and a rubric for your report are both in the diffusion module for Canvas for diffusion and in the Daphnia module for the Daphnia reports. Daphnia has 2 parts. The first part is due in lab this week. That's answering questions from the lab as well as extension questions. And then finally, there is going to be a second lab report, but that's not going to be due until the week of November 2nd in lab.